Hello there, and welcome to lesson 5 of cell biology. This one's on chromosomes and mitosis. Please make sure that you have pen and pencil, highlighter, ruler, all the tools that you need in order to make all the appropriate annotations. And do make sure that you are in a quiet space because you will have to concentrate quite a bit for this one. It's a bit of an information-heavy lesson today. But that being said, we're going to start off with our do now. So if you can make sure that you answer all 8 questions, I know I only have numbers 1 to 4 on the screen right now, but there are 8 questions. So answer all of them, please, and we'll go through the answers together. Perfect. So number 1, identify when cell differentiation occurs in plants. So this one is going to be from the last lesson. Plant differentiation occurs all the time, whereas in adults, it actually stops after when you become an adult. Your differentiation doesn't become as potent as it was before. So with plants, there's a part called the meristem, which is the very tips of the plants where most differentiation occurs, but they can always revert back to the differentiation cycle. Number 2, state where differentiation occurs in plant cells. So in animal cells, it's a little bit different. For animal cells, differentiation will occur in the embryo or during development. So if we're talking about a human being, when they're still a child, they still develop, which means that they have to have differentiation in order to grow the certain cells that they need in order to become an adult. So that moves on to number 3, which was kind of basically a statement saying what was the last lesson on. What's the difference between differentiation and specialization? So if you don't have the exact same answer that I have, don't worry, just as long as you have something along the same lines. So differentiation is the process of non-specialized cells turning into specialized cells, right? That's the actual change. Whereas specialization is actually having the separate jobs for each type of cell in a multicellular organism. So if we take a human example, we have nerve cells, we have red blood cells, we have muscle cells. So all of these different types of cells started off as stem cells, but then they went through differentiation to become specialized. Okay, so make sure you know the difference between the two. Number 4, identify 2 adaptations of a red blood cell. So I mentioned one last lesson, but I'm introducing a new one, which is red blood cells contains a pigment called hemoglobin. Okay, not hemoglobin, hemoglobin, that allows them to carry oxygen. All right. So basically, the more hemoglobin a red blood cell has, the more oxygen they can carry. And the other one is that hemoglobin has no nucleus, which gives it more space to store more hemoglobin, which will allow it to carry more oxygen. Okay. So please make sure you have all the answers there, and we'll move on to the next 4 questions here. Number 5, identify the number 2 adaptations of a xylem cell. So again, we're looking at plant cells again, and I did mention a couple of these. So one adaptation of xylem tubes is that they're able to form long, continuous, hollow tubes, and that's because the end parts of the xylem cells will kind of form together to create one long tube, and they're also fortified with a substance called lignin, which just like the name, just like it says, it strengthens and it supports the stem. Number 6, state what a unicellular organism is. So just breaking apart the word, uni means one, cellular means the cell. So it's an organism that consists of only one cell. So number 7 asks, what is a eukaryote? So eukaryote is going to have, is an organism that has its organelles enclosed in the membrane. And just being a little bit more specific with that, the nucleus of a eukaryote is always enclosed inside of a membrane, whereas in a prokaryote, the prokaryote does not have a nucleus, so the genetic material would be just kind of floating around in a cytoplasm for the prokaryote. And the last one is give an example of a prokaryote. So I gave a specific and a general answer. If you put bacteria, that's completely okay. I put E. coli down just as a specific example of what a prokaryote could be like. Okay, so please make sure that you have all the correct answers on your booklet, and we'll move on. All right, so today we're looking at chromosomes and mitosis. So hopefully by the end of today's lesson, you'll be able to describe to me what a chromosome is and where it's found. as well as describe where and how body cells divide, and also describe the diagram of the cell cycle and all of its stages. I know it seems like a lot of information, but I'm going to try and break it down as simple as possible, and I'm also going to be providing you with a couple links to videos on YouTube that will help kind of explain this a little bit further with animations and such. Right, so let's look at chromosomes first. You may or may not have heard the term chromosomes before, and if you did, that's fine. If you didn't, don't worry, it's not as complicated as you think. So let's start off reading in line 6 here. What makes you, you, is your DNA. It is individual to each of us, unless you have the luck of being an identical twin. However, what is DNA and where is it found? So let's start off on line 8. Deoxyribonucleic acid is made of phosphorus, carbon, hydrogen, oxygen. In the topic of inheritance, you will learn much more about this. All right, so I'll say that again, deoxyribonucleic acid. Okay, I like to break it down, deoxyribonucleic acid. All right, so highlight that sentence right there. And this will become important in the future when you learn exactly how DNA works in your body. And like it says, we're going to study much more on DNA on the topic of inheritance because that's what DNA is. It allows us to pass on our genes to the next generation, basically. So that's what DNA is. It's just basically your genetic material. It's what makes you, you. So where is it found? DNA can be found, this is at the end of line 9, it can be found inside the nucleus of a eukaryotic cell, and long lengths are condensed and coiled into molecules called chromosomes. So I want you to highlight that whole bit right there. So we can find it in a eukaryotic cell. So remember that in a eukaryote, the nucleus contains DNA and it is also enclosed inside of a membrane. It's not just floating around. DNA just doesn't float around in the cytoplasm unless you're a prokaryote. But humans are eukaryotes. All right. So then when you have a bunch of DNA, DNA kind of comes in like long strands, like a string. If you have lots of DNA and they're condensed and you coil them up, then we call that a chromosome. All right. So there's that word right here, chromosome. And I'm going to show you a picture of what chromosome looks like. But just before I do that, just be aware that chromosome comes from the Greek terms chroma and soma, which stand for color and body. So literally it means colorful body. All right, so I'm going to show you a picture right here. And you might notice the top part of like the stringy bit, that's kind of what you envision DNA to be. Whenever you watch cartoons or a TV show and you see that picture, you should think about, oh, genetics, right? So DNA, which stands for deoxyribonucleic acid, what we call a double helix structure. So it basically has two strands intercrossing every once in a while. And it also has these things called base pairs that attach the two strands together. Okay, so there's your DNA, and then it leads into this large structure called a chromosome, which is that large X that you see at the bottom there. So a chromosome is literally, think of all the strands of DNA coiled up into really, really small bits, and it's condensed, so it's really thick. All right, so that's your chromosome. For the other parts, you don't really need to know anything else except the part where it says centromere. So centromere kind of sounds like the word center, which is fair because the centromere is the middle of the chromosome. So where the two lines meet for the X, that's where your centromere is. And it'll become important in a little bit here, okay? So just keep that in mind. In humans, I'm continuing at the end of line 11 here. In humans, each nucleus carries 23 pairs, so 46 in total. Right? Pairs means two. 23 pairs, chromosomes. All right, so I'm going to show you a picture again. So you might see an image on your booklet that's similar to this, but this is basically all the chromosomes in your body. All right, you have 23 pairs, which means if you count them out, there's 46 X's in total. And these are basically what give you the definition of being you. All right, so if you took yourself and a friend, right, both of you would have different chromosomes. You'd have the same number of chromosomes, but it's really the reason why you two looked so different. All right, and so how do these chromosomes come about? All right, so I'm going to highlight the next bit here. So one of the chromosomes come from your mother, and one of the other chromosomes come from your father. All right. So when before an egg is fertilized, an egg will have 23 chromosomes, and a sperm cell will have 23 chromosomes as well. And when they meet, they basically combine all their chromosomes together to create 46 in total. And from there, the cell will start dividing and multiplicating, which then a fetus will appear. Okay. So there you have it. So there you have 23 chromosomes right there. And I want to focus on this one word right here where it says in brackets, leading to variation in individuals. Okay. So variation, you will go into depth when we talk about inheritance, but I want to introduce this topic to you. Variation is the difference between living things of the same species. Keyword being same. All right. If you were to compare a dog and a cat, yes, you would understand that they're very, very different. But if you compare two dogs of different species, say for example a golden retriever and a poodle or a chihuahua, all these dogs are very different, but they're all dogs at the end of the day. So what makes them different? The answer to that is basically their chromosomes. All right. So a certain chromosome might tell the cells to grow curly hair or flat hair or long hair, and these are basically what make us so unique. For humans, we have 23 of them, and each one of them codes for different things. So your eye color is heavily dependent on your genetics, your hair color as well. Everyone has a natural hair color. So, for example, if both of your parents have really dark hair, then the chances of you having bright hair is extremely small. All right, and there's always a chance because we call this a mutation. Mutations are basically things that happen in the DNA replication process that isn't really supposed to happen, but it happens anyways. Okay, so moving along. Right, so moving along here. So this picture is actually in your booklet, and I wanted to focus on the box on the right. Okay, and there's a word here that I want to focus on, and that word is karyotype. So you can see the sentence right here. So if I read out the sentence, it says, each chromosome contains a large number of different genes, the part that give you your characteristics such as eye color. The entire chromosome for an organism can be referred to as a karyotype. So if I were to ask you the question, what is the karyotype of a human being? Then you would show me a picture of this. All right? And this is basically what we call the entire chromosome, right? So from here, the karyotype of a human being, I can instantly tell there are 23 chromosomes, okay? And I can see the size of the chromosome as well. So you can see that number 1 is the biggest going down to the smallest, which is 22. The 23rd chromosome, where it says XX or XY, is called the sex cell. And this basically determines what sex you are, male or female. And if you have an XX chromosome, then you are female. If you have an XY chromosome, then you are male. All right? And you'll notice on each of the chromosomes, there's a little black line here and there. So these dark bandings represent a specific gene. So you might have, for example, if I were to say, chromosomes 4, 6, and 12 code for blue eyes. Okay, so then if you have blue eyes, you would look at your 4th, 6th, and 12th chromosome, and you could see exactly that the chromosomes fit for a person who has blue eyes. All right. Now, sometimes there can be abnormalities in the karyotype where there are more or less than chromosomes than the 46 than you originally have. So this, I'm going to have you underline karyotype here. This leads me to a condition called Down syndrome. All right. Now, you may have heard of Down syndrome before, and you may know someone who has Down syndrome. But basically, I want to show you what happens. Down syndrome is also known as trisomy 21, which refers to the 21st chromosome. So chromosome 21 should only have 2 chromosomes, okay, as with all the other chromosomes. But Down syndrome has an extra chromosome, which is the 21st chromosome. And depending on the severity of it, you can determine whether or not it has a full extra chromosome or maybe part of an extra chromosome or just a little bit. All right. But that gives you a basic idea of what could happen if you have an extra chromosome. So there are other conditions where if you are missing a chromosome, you will have a different condition as well. Okay? All right, so that is basically everything that we're talking about with chromosomes. I'm going to show you, like I said, I'm going to show you a couple links at the very end of the lesson to show you different videos that you can watch in order to clarify a couple things here. All right, but now we're moving on to mitosis. And before I even get into mitosis, I have to explain this first part. On line 2, it says, cells within an organism are always at a point within a cycle, which is called the cell cycle. All right, and that cell cycle is on the picture in your right here. So this cell cycle has two main parts. The first part is called growth and replication. And the name for this is called interphase. All right. The next part is called mitosis. So it really depends on how your teacher teaches this, but I like to include interphase in the cell cycle because you're going to learn the 4 other steps of mitosis, and in total you'll have 5. So the reason I have the 1 right next to mitosis is not for mitosis, it's actually for interphase. So interphase is the first step in the cell cycle. So what happens in interphase? I'm going to read along in line 5 here. During interphase, when it is not dividing into 2 cells, the cells grow bigger, increasing its cytoplasm and subcellular components, or organelles, such as mitochondria and ribosomes. It duplicates the DNA, doubling the amount of genetic material, and prepares for the cell to undergo mitosis. So mitosis is basically cell division. The cell divides into 2. If you have 1 cell, it becomes 2. Then the 2 become 4. Then 4 becomes 8. Then 8 becomes 16, and so on and so forth. But in order for all of this to happen, you don't want to have one set of DNA and just keep dividing it in half and half and half and half. Because once you do that, then all the cells at the very bottom are, when you have the 16 cells, are going to have a fraction of what DNA they're supposed to have. So you want to make sure that all 16 of those cells have the same amount of DNA as what you started off with. Okay. So that being said, interphase is the part of the cell cycle where everything duplicates. You're basically creating a backup for everything so that you're going to send your backup into the new cell and you have 2 cells that have a full set of organelles and DNA as well. So mitosis is going to be when a single cell reproduces by itself, forming 2 identical daughter cells. So we just call them daughter cells because they are the generation next. And then we call the first cell the parent cell. Now, the thing about these 2 daughter cells that form is that they're identical. If you look at the diagram on the right, you'll notice that the first cell and the 2 final cells are exactly the same. All right? And that's the key feature of mitosis, is that they're going to form 2 identical daughter cells. All right? It is one continuous process that is split into different stages. So I have the numbers 2, 3, 4, and 5. Because these are the 4 steps of mitosis, the first step being interphase. Okay, so now you have a total of 5 that we're going to learn. So in prophase, the chromosomes condense and the nuclear membrane disappears. Spindle fibers move to the opposite sides of the cell, attaching to centromeres. So remember, centromeres means the center of the chromosomes on the chromosomes. All right. Now, the picture on the right, I'm not going to lie, is not my favorite, but you can't really pinpoint these stages somewhere. So the prophase stage is somewhere along there where you have the spindle fibers moving to opposite sides of the cell. So they don't want to be next to each other. They want to be on opposite sides of the sphere. Okay. And the nuclear membrane is disappearing. So all that DNA that is stored inside the nucleus is starting to spread out. All right. And so that's all over the cell now. It's kind of like a prokaryote. So then the next step of it is going to be the metaphase. All right, so metaphase right here, step 3, is when the chromosomes line up on the equator of the cell with spindles at opposite ends. So the biggest difference here is that the chromosomes are now lining up along the center of the cell. Okay, and from equator comes from like the geographical term. Equator of the Earth is the center line around it, right? So same thing, you have chromosomes that are lining up at the center of the cell. Okay. And if that's the equator, then the spindles would be basically your north and south pole at the very top and bottom. Okay. All right. So now we have metaphase. Next step is anaphase. And this is where the centromeres will snap. So remember, centromeres are the center of the chromosomes. And basically what happens is the spindles are like little pieces of rope that are pulling on each side. Okay, so you notice in the picture that the chromosomes are kind of being pulled in two directions. All right, so anaphase is when that basically snaps apart. Okay, so now you have chromosomes in two separate sections of the cell. And this is where the cell starts to look like it's about to divide. Okay? Which brings me to the last phase, which is telophase right here. So this is where the chromosomes decondense and the nuclear membrane reforms as the cytoplasm divides into two and two identical cells are produced. All right, so this one I can kind of place it in this area because you can see that there's two identical cells and the chromosomes are starting to be decondense, and the nuclear membrane is starting to form back into a nucleus again. Okay? So there you have it. You have 5 stages: interphase, prophase, metaphase, anaphase, and telophase. Now you might be wondering, well, how am I going to remember all these stages and exactly what they do? So what I can do is I can help you memorize the stages in order of what's happening, okay? So this is a very simple mnemonic device that you can find on the internet, and the letters represent the different stages. So IPMAT stands for I picked my apples today. So each letter represents a different stage: I for interphase, P for prophase, and so on. And I like this picture better because it actually shows you the spindle fibers in the diagram. So this one is labeled as mitotic spindle, but you can see that the spindles are attached to the chromosomes and they pull apart each chromosome so that you have identical chromosomes at the very end. Now you notice at the very end there it says telophase and cytokinesis. Cytokinesis is technically a part of mitosis, but it's one of the lesser known ones. But if you're really curious, cytokinesis is the part where the cell actually divides into two. Okay, that's all it is. So there you have it, a simple tool for you to use. Right, so the last part I'm going to explain is the links that I promised you that will lead you to different parts of YouTube where you can see some more informational videos that might help fill in the gaps. So the first one is actually in your booklet, right under where it says learning activity. So that YouTube link leads you to an incognito video talking about the cell division or the cell cycle. The next one that's in the box of the checkpoint where it says tinyurl.com/y2mkoamt. This one will lead you to a video on chromosomes and also mitosis. All right, so both of these videos will help you see for yourself what is going on with the whole division process. And it does help to see videos on this because obviously I can't do animations, but these videos can. So for this question here, where it says describe the main steps in mitosis, it's worth 4 marks. And if you've been following along with my lessons, I keep telling you that the marks represent how many different points you should tell me. So in mitosis, you know that there's 4 steps, okay? There's, so think back to the I picked my apples today, right? So there's, prophase, metaphase, anaphase, and telophase, right? So basically you would get 4 points for saying those 4 stages and describing them to me. All right, so there's your 4 marks. Right, so that brings us to the knowledge check at the end of the lesson. If you want to pause the video right now and get started on the first 8 questions, and then 9, 10, and 11 we'll do together. Perfect. All right, so number 1, identify what a chromosome is. So going back to this lesson, a chromosome is basically all that condensed and coiled up DNA, or you could have put genetic material as well. Either or is fine. Number 2, state the literal Greek translation of chromosome. So the word chroma comes from color, and soma comes from body. So if you put them together, we just get colored body, or a color body. Number 3, state the name of the growth and replication stage. So this one would be before mitosis, which is called interphase. Number 4, all cells carry out mitosis at the same time, true or false? This one is going to be false, just because mitosis is an entirely separate step from interphase. And if you go back to the picture of the little circle, that shows you how much it's in interphase and how much it's in mitosis, you'll notice that in a cell's life or in a cell cycle, a cell will spend 3/4 of its time in interphase rather than mitosis. Okay? And also, not all cells go at mitosis at the same time. All right, number 5. State how many cells are produced during mitosis. So, any one cell that goes through mitosis, how many do you get at the very end? And the answer to that is 2. Number 6. Describe those cells. So, 2 cells that are produced at the end of mitosis, what do they look like? Or just describe them. And they are both going to be identical to the original cell. Or you can put parent cell. I don't know if I mentioned it like that, but they're both, since the 2 cells are called identical daughter cells, you would call the original cell the parent cell. Number 7, identify what a karyotype is. So going back, a karyotype is the entire chromosome for an organism. If I asked you what's the karyotype of a human being, you would tell me 23 pairs of chromosomes, or you can show me a picture of all 23 chromosomes. Sorry, all 23 pairs. Number 8, identify what DNA stands for. So DNA, very simple, stands for deoxyribonucleic acid. Okay, so those 8 questions, please make sure that you can answer them quite easily because this is the meat of today's lesson. For 9, 10, 11, it requires a little bit more thinking just to figure out, that extra step. So number 9 asks, suggest what would happen in the cell cycle if your DNA did not replicate. So basically, if your cells did not go through interphase and it just went to mitosis, your daughter cells would not be identical, right? Because you don't have the same amount of DNA. So actually, the chances of those daughter cells surviving are very, very low. Okay? We're going to move on to 10 and 11, but make sure you have those answers copied down. And number 10 asks, suggest what would happen in mitosis if your chromosomes did not line up down the middle of the cell. So in one of the steps, remember that the chromosomes will line up along the equator of the cell, okay? And this is where the spindle fibers will attach itself. But if the chromosomes did not line up, so they were just kind of randomly out throughout the cell, then what would happen in mitosis? So, if you can imagine, then one daughter cell might have more chromosomes than the other. Okay? So the reason why they line up is so that you ensure that both daughter cells have the exact same amount of chromosomes. Number 11. Suggest what would happen if the subcellular subcellular structures or organelles were not duplicated enough during interphase. So going back to those first lessons where we talked about organelles, you should remember that a cell has many organelles and many of the same type of organelles. So for instance, a cell will contain lots of mitochondria in order to produce energy. But let's just say that it wasn't duplicated enough, so now you don't have enough mitochondria. Then what happens is one daughter cell might be missing certain essential organelles if most of those mitochondria were split onto one side. Or it might happen the other way, where you do split the mitochondria evenly, but both cells now don't have enough to survive, and the chances of them surviving drop significantly. All right? So just make sure that you have all the answers, and if this lesson is confusing, like I said, go back to those 2 videos, or 2 links to YouTube to just see a little bit more extra information on mitosis. It is one of the more difficult subjects in biology, but it's also one of the most commonly taught. So you will be able to find many, many resources for this topic here, okay? And thank you again for watching.